wash the well, and then stain with crystal violet to indicate biofilm formation. And so if we think about where GBS comes from and where it goes during these infections, starting in the gastrointestinal tract, there's some micronutrients that would be very different in these two environments, vaginal canal, amniotic fluid, compared to the gastrointestinal tract. One of these is glucose. Glucose is in fairly low concentration in the colon, but within vaginal fluid and the amniotic fluid would be relatively higher. So if we look at several different strains of GBS, and we supplement the media with just a little bit of glucose, we see a significant increase in many of these strains as far as its biofilm formation. So the biofilm is producing based on how many cells were present. Another micronutrient that we have interest in the Gaddy lab is zinc. So zinc is present in very little amounts in vaginal fluid, be relatively higher in the gastrointestinal tract. So if we grow GBS in the presence of increasing concentrations of the zinc chelator, so again, this T-pen will bind up zinc so it's no longer available for the bacteria to use. As we increase the amount of zinc, we see an increase in the amount of biofilm shown in the crystal violet stain. It's quantified below. You can see this under scan electron microscopy. A little bit of biofilm forming in medium alone. We add T-pen to remove the zinc would be available for bacteria. You have this dramatic increase in the size of the mushroom clouds, and again we can see those nutrient channels of these biofilms. Now if we add back zinc chloride to just overwhelm the system so that zinc again becomes available to the bacteria, we lose that biofilm formation, and we see that here. So these different micronutrients may drive changes in GBS physiology that make them more prone to colonize the vaginal epithelium and potentially to ascend, but that's something we're still working on. The other part of this is I'm going to move into some emerging diagnostic work. Because we've shown that these bacteria form biofilms on the surface of the fetal membranes, we partnered with a lab in biomedical engineering that's really interested in identifying bacterial infections within tissues. And so with Oscar Olea, we looked at different GBS strains on agar. You can see that there's very clear distinctions between some of them. GB1084 is very pigmented. That's that beta hemolysin, that pore-forming toxin that it produces, whereas some other GBS strains like GB37 make very little of that pigment and therefore are not very orange. And then we have some strains that are intermediate. If we compare that to just the coloration of MRSA and E. coli, we can notice there's a difference. Now this lab uses Raman microspectroscopy to try to identify bacteria as well as infection. And what Raman is when light hits a surface like the table in front of you, a lot of it's reflected, but there's a very small amount that's absorbed, and it's changed. It's given off at a slightly different wavelength. This can actually be detected and is known as the Raman shift by this Raman microscope. And it allows us to potentially identify differences within tissue, so it detects changes in macromolecules like carbohydrates and lipids and proteins. And so if we look at the Raman spectra of these different GBS isolates, you can see there's some commonalities among the isolates, but there's at some points subtle differences, and sometimes rather large differences. So in GB1084, we have these large peaks at 1120 and around 1540. This is directly related to that pigment production that the Raman spectroscopy can identify. Some of our lesser pigmented, like GB2, we see smaller peaks, and then we see different peaks in both Staph aureus and E. coli. Staph has a different carotenoid pigment that it produces. And so we wanted to see, based on these spectra, we group or identify what's GBS and what's not. And so we used hierarchical clustering, and you can see that the majority of our GBS strains actually cluster together, as opposed to Staph aureus and E. coli, which break off, and then we have one outlier, GB1084, because of the intensity of those peaks. So all we've done is look at the bacterial cells on agar. The real goal of this was trying to see, could we identify infection within these fetal membranes? So we go back to our transwell model. I've infected these tissues either with GBS, 3 different strains that have different pigment production, 1084, 37, or 590, E. coli, or MRSA. And basically, when we looked at these spectra, again, there are some similarities, but there are also differences. Can we identify those differences that are important to potentially diagnose infections in these tissues? And if we use a machine learning algorithm, and that's what these black bars are, it can assign importance to these different peaks. And when we do that, could identify 51 out of the 55 clinical samples that we then infected ex vivo with GBS. And then we can then confirm that GBS is present at the site of our measurements because we make these small cuts within the membrane tissues. And again, there are thick biofilms that form at 48 hours of infection that we can measure. So we've talked about what the bacteria may be doing. We talked about ways that we could potentially diagnose these early changes and potentially diagnose infections in vivo over time as we grow kind of more sensitive with these diagnostic techniques. I want to shift back and look at the host immune response to infection. So if we infect these tissues with either GBS or Staph aureus, two different bacterial pathogens, you can see that the type of inflammation we get is slightly different, but the hallmarks, I think, are the same. That we have pro-inflammatory mediators, including IL-6, tumor necrosis factor, IL-1 beta, that seem to be released by these tissues themselves. And that's important because, again, these are the kind of mediators we find in the amniotic fluid of those women with intrauterine infection. So the membranes themselves can recognize bacterial pathogens. And a lot of these cytokines will be predicted to cause the infiltration or trafficking of phagocytes into these tissues. So is that what we see? So if we look both at the human fetal membrane model as well as the mouse model, just at the gestational or fetal membranes, we can again identify areas of GBS infection using immunohistochemistry, and then we can identify macrophages, CD868 positive cells at that same location. And again, these are macrophages, and this is a disconnected system. So if we then look at the mouse, we actually have an in vivo system. We see again, GBS at 48 hours of infection in our mouse model causes a pretty robust infection of these tissues. Again, we can find F480 positive macrophages at the site of infection. So macrophages are present at the site of infection, and we'll come back to that in a second. I want to talk briefly about the mouse model again. So after we do that intravaginal inoculation, what happens with GBS? Well, over 48 hours, we can actually see broad dissemination into the gestational tissues. We can isolate GBS from the vaginal canal, but as well as the uterus, the placenta sample, all the way to the fetus. So GBS broadly disseminates in this model. And we can show that also by immunohistochemistry. So if we take one of the fetal placental units, so here is the pup, here is the placenta, and you can see this brown staining where GBS is present, both in the decidua, this outer layer, as well as the junctional zone of the placenta. We see less invasion into the labyrinth of the placenta at 48 hours. If we look at the H&E at those sites of infection, we also see these polymorphonuclear cells, or neutrophils, present, and then we can quantify those using flow cytometry. So again, we see, compared to those animals that are uninfected, if we infect with GBS at 48 hours, we see this infiltration of neutrophils into these tissues, both the decidua and the placenta. And again, this is important because this is what happens during chorioamnitis in people. So this inflammation, this neutrophil invasion into these tissues. So we've looked at macrophages. What about, neutrophils, what about macrophages within this placental tissue? And again, at the areas of GBS infection, we can see F480 positive macrophages present. There are some present in uninfected tissues, and the staining is a little difficult because this tissue is so necrotic. So just to check back in, what have we talked about so far? If we focus on the fetal membranes during infection, what happens? GBS and other bacteria are able to form biofilms on the surface of the choriodecidua, and this has been shown from clinical samples as well, that this happens and biofilms form on the fetal membranes, and GBS in particular can invade. In response to that, the fetal membranes release cytokines that leads to trafficking phagocytes into these tissues. And now I want to zero in a little bit more on the macrophages, one cell type of this multi-cellular structure. And so in order to study placental macrophages or Hofbauer cells, these are fetally derived macrophages, how do we do this? Well, we go back to our clinical samples that we get, and we basically isolate placental macrophages from these tissues. We do this by mechanical enzymatic digestion, putting them through a density gradient and then cell sorting, and we get cells that look like this. Because we talked about the different things that these cells do during normal pregnancy, we wanted to look to see how this changes in the context of an infection. And so we started by, again, a simple experiment. Let's use our high-resolution microscopy techniques to look at the host-pathogen interface. How are these two actually communicating in setting an infection? And we hypothesized that there would be morphological changes or structural changes that happen in these cells. What we weren't so prepared for was some of the changes that we saw. And so when looking at these cells under scanning electron microscopy, I saw several cells that released these net-like structures, or these webs. And they looked like they were coming from the cell. And so if we look at higher power, you can see these very fine fibers pulling off of the cell body and extending into this web-like structure. And this GBS that I've infected in the plastic dish actually is getting caught and wrapped up in that structure. So what is this? We thought this was very reminiscent of neutrophil extracellular traps. So these were first described in 2004. And it's a unique cell death and immune response that was first described in neutrophils. And what's thought to happen is that you have neutrophils upon some stimulus basically degrade their nuclear and granular membranes and mix those contents together and shoot them out into these cellular extensions that are composed of DNA, as you can see in the staining here. With that DNA that's shot out of the cell, you have histones and those DNA binding proteins, but as well as granular proteins like elastase. And they showed this by both immunofluorescence and immunostaining for histones and elastase. Now, the original description of extracellular traps in neutrophils or nets talked about how these structures could trap microorganisms and potentially be bactericidal. And they do this because you have these proteolytic enzymes in high concentrations right adjacent to the bacterial cells. And so I went to then figure out, were these extracellular traps being produced by macrophages or METs in these cells? And so the first study we did was just looking at, if we use a DNA binding dye, a DNA intercalating dye, what do we see under a confocal microscope? And you can see in those uninfected cells, we have a few projections coming off cells. And often these projections are several times the length of what a normal cell would be. If we infect cells with GBS ex vivo, we see many more. Now, the staining itself suggests that maybe these structures are composed of DNA. But if we treat with the DNAs, we lose all those structures, okay? Suggesting that the backbone of these cellular extensions is indeed DNA. And we see similar responses if we look under scanning electron microscopy. Again, long extensions that disappear when we treat with DNAs. If we quantify that response, we see just 1 hour of infection. So this is an MY of 20 to 1, 20 bacterial cells to 1 macrophage. We see about a 2-fold increase in the number of extracellular traps in just that 1 hour. Not only that, but if we increase our MOI, increase the number of bacteria we use, we see a dose-dependent response.